For a long time, we pictured the inside of a neuron as relatively still, a simple wire transmitting electrical pulses. But in the 1940s, a biologist named Paul Weiss decided to test this. He gently tied a thread around a living sciatic nerve in an animal, constricting it like a kink in a garden hose. He waited. What he found when he looked under a microscope days later changed neuroscience forever. On one side of the thread, the axon was swollen and bloated, while the other side had withered. Something was clearly trying to flow down the nerve, and he had created a dam. This damming experiment was the first real proof of what we now call axonal transport. The neuron wasn't a static wire. It was a dynamic, flowing highway. This cellular goop, the axoplasm, was in constant directional motion. But what was it carrying? And more importantly, what was the engine driving this microscopic traffic? The answer lies in an incredible internal skeleton that gives your cells their shape. Inside every one of your axons is a network of ultra-strong protein filaments called microtubes. You can think of them as railway tracks, laid down from the cell body all the way to the farthest tip of the axon. These tracks are incredibly long and stable, providing the infrastructure for a logistics network that dwarfs anything humans have built. Every single signal your brain sends depends on the integrity of these molecular rails. If microtubules are the tracks, then what are the trains? Meet kinesin. Kinesin is a motor protein, a molecular machine that literally walks. It has 2 feet that step along the microtubule, one after the other, burning cellular fuel, ATP, with every step. Clutched in its hands is a bubble-like container called a vesicle, stuffed with precious cargo. This kinesin-driven movement, from the cell body outward to the synapse, is called anterograde transport. It's the outbound delivery service. What's in the packages? Everything a distant synapse needs to function. Mitochondria to act as power stations, neurotransmitters to send signals, and the literal nuts and bolts, proteins, to repair and build the synapse itself. Your ability to form a new memory is directly dependent on these tiny delivery trucks reaching their destination. But traffic on this highway runs both ways. For the return journey, a different motor protein takes over. Its name is dynin. Dynin walks in the opposite direction, from the synapse back towards the cell body, a process called retrograde transport. It's the cellular recycling truck and information courier. It carries back used-up components for breakdown and recycling, preventing a buildup of cellular waste at the synapse. This return trip also carries critical information. Dynin ferries chemical messages from the synapse back to the nucleus in the cell body. These messages are essentially status reports, telling the cell's command center if the synapse is healthy, if it's active, or if it's been damaged. The cell body then uses this information to decide which new cargo to ship out via kinesin. It's a constant, dynamic feedback loop. How fast is this traffic moving? It varies. The fast lane of axonal transport can move cargo at speeds of up to 400 millimeters per day. That might not sound fast to you, but on a cellular scale, it's blistering. It's the equivalent of a person walking from New York City to Philadelphia in about 20 minutes. There's also a slow transport system, moving larger structural components at a more leisurely pace of just a few millimeters per day. This entire system is mind-bogglingly crowded and complex. A single axon can contain thousands of microtubules, each one a potential highway for countless kinesin and dynin motors. They are all moving at the same time, carrying different cargo, somehow avoiding collisions and delivering their packages to the correct locations along a nerve fiber that can be over a meter long. Imagine every package delivery service on Earth operating simultaneously within a single microscopic tube. What happens when this system breaks down? The consequences are devastating. Many neurodegenerative diseases, like Alzheimer's, Parkinson's, and ALS, are now linked to defects in axonal transport. You get molecular traffic jams. Cargo doesn't arrive, waste products build up, and the neuron's distant synapses slowly starve and die. The cell, cut off from its own supply lines, withers. So, the image of the brain as a static network of wires is profoundly wrong. It's a fluid, ever-changing city. Its streets are microtubules, its vehicles are motor proteins, and its cargo is the very substance of thought and memory. Every second, millions of these microscopic deliveries are successfully completed inside your head, rebuilding, refueling, and reconfiguring the connections that make you, you. This isn't just maintenance, it's creation. The tissue in your brain is never the same from one moment to the next. The proteins that make up a synapse today might be recycled and replaced by tomorrow. This constant movement is the physical basis of learning and adaptation. When you learn something new, you are initiating a cascade of these transport events, physically altering the structure of your brain. You are not just thinking, you are literally becoming a new city. Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.